Hello viewers and students. Welcome to the program Myself and My Bouskein. So in today's program we have a great delegate Mr. Karthikeyan sir. So let us welcome sir with a great round of applause. Hello sir.
Hello, ma'am. Greetings
So
to all of you.
welcome to our program Myself and My Bouskein show.
My pleasure, ma'am.
So in today's program sir, some of the viewers and students
Yeah.
they are waiting for some of the answers to the questions.
Yes.
So please, we
Please.
would like to ask some questions to you, so you could please answer to those.
Yeah, definitely. So, respectable viewers and my dear students. So, you have asked a few doubts in the form of questions. So, I am here now in this video. So, to clarify your all doubts and questions. So, please let's watch.
So let's get started.
Yeah, please.
Please, we have a first question. What is biotechnology?
Biotechnology. So nowadays frequently we are listening this word. So this is familiar word in our regular life. So bio in the sense life you knew already. So the technology. So technology nothing but so especially these microbes. So the microorganisms and their properties and their cell structure and their program and the content whatever present in them. So these microorganisms technically used by our humans for our life and our lifestyle and our upcoming future sake is known as biotechnology. So this is a type of science.
Yes, sir. Then we have a next question. What are molecular scissors?
Molecular scissors. So this is a task from molecular biology. So here scissors in the sense we knew already the special instrument which is responsible to cut something, to separate or divide something. So here molecular scissors in the sense especially enzymes, nothing but a catalyst. So here DNA, deoxyribonucleic acid. So in this DNA special sequences are there, special codons are there, nucleosides and etc. So in a DNA technology, especially in genetic engineering, so to cut and separate those sequences at the particular site, special type of enzymes are required. So these are known as molecular scissors. So their functioning is cutting or separating the bonds or the particular site in a DNA strand.
So you mean the enzymes are called molecular scissors,
Yes, the enzymes
which are
especially.
useful for breaking or separating the DNA fragments.
Fragments, yes.
Yes,
DNA
very nice.
strand into fragments.
Okay, sir. The next we have a question. What is eco-RI?
Eco-RI, nothing but the simply we shall call that as E. So you knew, I think so, you are familiar with the bacteria E. coli, Escherichia coli, which is present in a human small intestine and it is used as a vector because it has a special circular DNA is known as plasmid. So here E. coli is nothing but that is also a type of restriction enzyme. This is obtained from the bacteria named as E. coli or Escherichia coli. This E. coli function is it will cut or restrict the bonds between GA in the sense guanine and adenosine. So we knew already thymine, cytosine, guanine and adenosine, ATGC bonding sequence. So here E. coli is responsible to restrict or cut between G and A, guanine and adenosine.
So you mean this is useful for restricting the bonds present between the pyrimidines and purines.
Yeah,
That is
yes.
nothing but a nitrogen base.
Exactly, exactly.
Very nice, sir.
Yes.
Then we have one more question. What are cloning vectors?
Cloning. So this term also familiar for you. In 8th standard itself, you might have been learned that Ivan Wilmot. So he was the first scientist who was cloned Dolly, a lamb. So here Cloning nothing but copying, doubling, multiplication. So here cloning vector nothing but same like chromosome is a hereditary vector. So some pathogens are vectors for spreading the diseases, same like. So here cloning vector in the sense, example plasmids. Plasmids and casmids are known as cloning vectors because the desired DNA is inserted into these plasmids, and these plasmids again injected into the body of the microbes, especially E. coli, and they become a double multiplication. So those are known as cloning vectors. This is highly useful in DNA technology.
So very nice. Next we have one more question.
Yeah,
That
please.
is, what is RDNA?
RDNA, or in the sense recombinant. So RDNA technology or recombinant DNA technology. So nowadays this is manual process by the humans, artificial but manual, nothing but common process. The selected or desired or foreign or source DNA is injected into plasmid, and the plasmid along with this inserted source or foreign DNA together known as RDNA. So this RDNA again injected into a microorganism body, nothing but E. coli, then the multiplication process will be forwarded. So this hybrid or artificial DNA is known as RDNA, nothing but recombinant DNA.
So you mean this RDNA which is formed will be the intermediate one before inserting into the original host body?
Yeah, this is the intermediate one. So later this should be inserted into the host organism. Yes.
Then next one more question we have. What is downstream processing?
Yeah, downstream. So this downstream processing is the last step followed by the scientist in the DNA technology. Again, DNA technology, it requires tools, DNA tools, around 5 to 6 type of tools it requires. Then downstream processing in the sense, after following all steps, the sample or the result or the product, it must be tested by the GEAC, Genetical Engineering Approval Committee. So before releasing into market, this product must be collected and checked, and finally it must be certified. Then only it must be released into the market for the further processes. So that step is known as downstreaming or downstream process.
So this step will be the last in the RDNA technology,
RDNA technology.
where they have to check everything, then they are going to release into the
Yeah,
market.
yeah. This is under the control of GEAC.
Well, then we have one more question
Yeah, please.
from a different viewer.
Yeah.
That is, how can we visualize DNA?
Generally, DNA is a nucleic acid. So it will be present the depth of this cell inside the nucleus. So DNA generally it contains it is negatively charged molecule. So to visualize DNA fragments, we must require gel electrophoresis technique. So in this gel electrophoresis technique, especially on the gel, agar gel, that is carcinogenic agent, we should not be touched, that very dangerous, highly. So on the gel, by adding a special chemical, ethidium bromide. So by adding this ethidium bromide chemical, we shall able to clearly visualize the DNA. And this is also a step in DNA technology.
So this process can be done before the downstreaming.
Downstreaming process, yes.
Then we have one more final question.
Yeah, please.
Like, what are endo and exonucleases?
Yeah. Just at the starting of the program, we have discussed about that nucleases. Restriction enzymes, molecular scissors. So here molecular scissors nothing but endonucleases and exonucleases. So these endo and exo, both nucleases comes under molecular scissors. Endo in the sense inside, depth. Exo means outside, not in depth. So these exonucleases are the restriction enzymes which they may cut the bondings or the DNA fragments at the end of the DNA. Then endonuclease is nothing but they may cut or separate the bonds within the DNA at the specific site. So one is end of the DNA and one more is at the middle of the DNA at the particular site, because one is called as exonuclease and one more is endonuclease. I hope you understood.
So very nice, sir. Thank you very much. You have answered all the 10 questions
Yeah.
which were given by our students and viewers. So thank you very much for giving this valuable information and explained in a very simple manner where all the students are capable to understand this. So thank you very much for being a part of our program, sir.
Thank you very much, Toonam. So you are conducting nice program that I mean myself and my Bouskein. It's a nice program. This is highly useful for viewers, especially students who are trying to approach and achieve NEET and etc. MSET also. So I am fortunate that to be a part of this program, that is myself and my Bouskein. So thank you, ma'am, and thank you, viewers and students. Myself, Karthikeyan, mentor in NEET. Thank you very much. Signing off.
I hope you all understood today's program. Keep watching. Stay tuned for further queries.